Well, good morning, everybody, and thank you so much for joining us on this rainy Tuesday morning with a lot going on the campus. This symposium is part one of a two-part celebration of the 5 members of the NIH family elected this year to the National Academy of the Sciences. Drs. Thomas Kunkel, Kyung-Kwan Chung, Giorgio Trinchieri, Sandra Wolin, and Stephen Rosenberg. So today we'll hear from Drs. Kwan Chung, Kunkel, and Trinchieri. And on November 13th, we will hear from Drs. Wolin and Rosenberg. So first I'll tell you a little bit about each of today's 3 speakers and then allow them to proceed with their inspirational talks uninterrupted. Hopefully we'll have time for a question or two after each of the talks. And please, if you do have a question when each talk ends, please use the microphones on either side of the auditorium. Dr. Kyung-Kwan Chung is a native of South Korea, and she received her undergraduate degree from Ichwa University, Women's University in Seoul. She received a Fulbright Fellowship to pursue her doctoral studies in bacteriology at the University of Wisconsin. In 1966, she joined NIAID and the field of medical mycology. She has been the chief of the molecular microbiology section in the Laboratory of Clinical Immunology and Microbiology since 1995. She received the Lifetime Achievement Award from the American Society for Microbiology in 2017, and her talk today is entitled In Step with Science, a Half-Century of Journey in Medical Mycology. Dr. Thomas Kunkel received his doctorate in 1977 from the University of Cincinnati. Following a postdoctoral fellowship at the University of Washington, he joined NIEHS in 1982. Dr. Kunkel currently leads the DNA replication fidelity group within the Genome Integrity and Structural Biology Laboratory. His lab studies DNA replication and how environmental stresses perturb this process. His talk today is entitled Accuracy Matters. Dr. Giorgio Trinchieri is a native of Italy who received his MD at the University of Torino. He joined the NCI Center for Clinical Research in 2006 and is currently chief of the Laboratory of Integrative Cancer Immunology. Prior to joining NCI, Dr. Trinchieri was the director of Schering-Plough Laboratory for Immunological Research in Dardilly, France, and an NIH Fogarty Scholar in the Laboratory for Parasitic Diseases at NIAID. And his talk today is entitled Immunity to Infections, Cancer, and in between. Congratulations to all of you, and Dr. Kwan Chung, the podium is yours.
Thank you. Can you hear me all? It's quite a challenge to talk about 55 years of career trajectory in 30 minutes. So I will start as roadmap for this talk will include personal background first, and then who are the mentors of myself. And then since I spent, I had about 70% of my work was in Cryptococcus, I will have a brief introduction to cryptococcosis. And then chronology of milestone research achievements and their impacts on the progress of medical mycology. What is milestone anyway? I define this as if that work laid the foundation so that the progress can be made in new direction in medical mycology. And then conclusions and acknowledgement. First, personal background and mentors. In 1960, I applied for a graduate study in the US because I received a Fulbright scholarship. And I was teaching at Ewha Womans University in South Korea, Seoul, Korea, because this was after my master's degree in bacteriology from that university. I was already a mother of 2 little boys at that time. And this person here is now, that time a toddler, but now he's a PI at NIH National Heart and Lung and Blood Institute. And he has PI, and he's working 3 floors down from my office. My PhD work started at the University of Wisconsin in 1961. If you want to figure out what is 1961 look like, this was 8 years after DNA structure was found, or at least became clear. So Watson and Crick's DNA structure, and 8 years after that. And 10 years before, the first endonuclease, restriction endonuclease became available. So first year graduate school was supported by the Fulbright, as I said, and the fellowship was managed by U.S. State Department. And the remaining years, the NSF research grant for 3 years, because Dr. Raper had an NSF grant to make a new monograph of Aspergillus. Then I completed doctorate in 1965. And I'm very sorry for this fuzzy picture, but Dr. Raper and I was standing in front of the building of bacteriology at University of Wisconsin in '61. Who is Professor Kenneth Raper anyway? Most of you may have not heard about him. He is the father of systematics in the most common mold genus like Aspergillus, Penicillium, and cellular slime mold. He's the one discovered the cellular slime mold. And he discovered very high penicillin-yielding strain of Penicillium chrysogenum, and Pfizer used this strain, and as a result, wholesale cost of 10,000 units of penicillin was $20 that became 3 cents by World War II because of his strain. He discovered and described the life cycle of cellular slime mold, Dictyostelium discoideum, in 1933, which became model organism for cell biology research. My career in mycology took off, actually, after the discovery of heterothallism in the genus Aspergillus, which I discovered while I was in graduate school. Because Dr. Raper gave me the research project saying, isolate as many new species of Aspergillus and describe phenotypically, biochemically, and so that was my project. And where was this Aspergillus coming from? It was coming from soil collected in Costa Rica. Why Costa Rica? Because most of the Aspergillus and Penicillium isolated from soil were from temperate zone, climate zone, and very little was known about those Aspergillus population in tropical soil. Because he got the NSF grant to make a monograph of genus Aspergillus, he had to include as many species as he could. And I discovered 9 new species that was isolated from Costa Rican soil. And then one of them turned out to be the first heterothallic species in the genus Aspergillus, which is composed of 132 species. Now, what is the heterothallism? That is describing the pattern of sexual reproduction in fungi. So, first type is homothallism. That's self-sterile and produce sexual state in pure culture. So there's no mating or anything like that. So in other words, it's a hermaphrodite. Second type of sexuality is called heterothallism. It's a self-sterile but cross-fertile. Now, it requires mating with opposite mating type and to complete the sexual life cycle. So I found this heterothallic Aspergillus from Costa Rican soil, and that really became the launching pad for my mycological career. Where did I get medical mycology training at NIH? Dr. Chester Emmons, who used to be the section chief of medical mycology at NIID, he is a legendary pioneer in pathogenic mycology in the United States. And he hired me as a visiting fellow in medical mycology section. And he discovered the ecological niche of almost every important systemic fungal disease, such as histoplasmosis, cryptococcosis. And then he's the one found the association of coccidioidomycosis with desert rodents. His 3 editions of medical mycology served as the reference for medical mycology in graduate school and medical school for more than 3 decades. To my disappointment, he announced retirement and gone after 6 months after my arrival. And so I was forced to function as a investigator on my own and didn't really get the benefit of being trained by Dr. Emmons. In 1974, I joined clinical mycology section headed by John Bennett in the Laboratory of Clinical Investigation. As you can see here, when it comes to diversity, 0. 0 diversity in race, gender, and academic degree. Everyone was MD, everyone was white male. And then I pointed myself with the yellow arrow. Can you see it? So my presence made it to LCID as a 98% homology instead of 100%. Well, it wasn't a very comfortable environment because I felt like an oddball. But the savior was John Bennett. He gave me the chance to build a program on Cryptococcus and cryptococcosis. And because when I joined, and he just opened his resources for me to use, he had nearly 1,000 strains of serotyped Cryptococcus neoformans in his stock. And then he had all the information about where this is coming from, whether it's a clinical source or environment source, if it is, what kind of a sample or clinical sample this isolate come from. So since I worked 70% of my career on Cryptococcus, I'd like to introduce briefly about the fungus and the disease. As you can see here, the disease starts from inhalation, and it will multiply in the lung if the individual has some immune defect issue. And then it will hematogenously spread and cross the blood-brain barrier and cause fulminating brain disease, which is really 100% fatal unless treated. And the risk factors, HIV positive AIDS patients are the highest risk. Idiopathic CD4 cytopenia and corticosteroid treatment or organ transplantation or even cancer. These are the risk factors for cryptococcosis. And its most common cause of death is meningoencephalitis. And actually, this is the most common cause of meningitis, not viral. If you remove the viral, this is really the most common meningitis cause in the US. Here I'm showing a very interesting looking brain, of course. The left, you can see the brain is covered with a white. This means the cryptococcus is growing very well on the mostly on meninges. And the other one is a section, brain section, and this glistening area means a collection of cryptococcal capsule as well as fungal growth. Now, this fungus is neurotropic because it has ability to cross the blood-brain barrier first, and then it can survive in very low glucose environment. And then it has enzyme called lactase, which oxidize dopamine, epinephrine, and then norepinephrine. That's sort of neurotransmitters. And now I'm going to show chronology of milestone research. As I said, milestone means if it laid the foundation for a new direction of study in Cryptococcus and cryptococcosis. If you look at this slide, the lower part is the development of technological development, which is relevant for our study. And then I highlighted the parts which are all cryptococcal research. And in the most important part is I discovered the heterothallism in 2 agents of cryptococcosis in 1975 and 1976. So this is a year after I arrived at the Bennett's laboratory. I asked him, can you collect, just pick 20 isolates from half clinical and half environmental from your 1,000 stock cultures. And he picked 20, and then I was thinking maybe it's a heterothallic and started from there. And then I formulated CGB media that can separate 2 cryptococcal pathogens, because that time, you remember, there was no DNA sequence or ITS or anything, and no Malditoff for sure. So for the identification of these 2 species was quite laborious, because you had to have either antibodies specific to each serotype, and you know how it cost and the time that's spent to get the antibodies. So this media really laid the foundation for lots of things, and then discovered most important basic virulence factors of Cryptococcus in '82. And then transformation of Cryptococcus, the DNA-mediated transformation in 1990, and then created a congenic set. And one of the strain that I created was used for the first genome sequencing strain of Cryptococcus. And once transformation technique was achieved, and first thing we did was molecular cause postulate was fulfilled in 1994. And we identified the first fungal quorum sensing peptide because quorum sensing material in fungi were all organic acid, and peptide was only found in bacteria. But we found the Cryptococcus has the first peptide quorum sensing material. And then azole heteroresistance, GM-CSF autoantibodies as a major risk factor for C. gatti. But I cannot go through all this, and I'm going to just concentrate on the first 5 subjects because that's really the major foundation we laid for the progress. So how did this work, which I call milestone work, lay the foundation for advancement of what field or what research in Cryptococci? First, taxonomy and phylogeny. Cryptococcus was not even classified properly because it didn't have a sexual state before. And then phylogeny of the fungal kingdom could be studied, including Cryptococcus. And pathobiology field, identification of essential virulence factors, and the fulfillment of molecular cause postulate for fungal virulence factor. And then ecology, identify the new source of ecological niche of Cryptococci. When it comes to epidemiology of the 2 species that cause cryptococcosis was quite different. And so epidemiology was progressed after this CGB media utilization. And genetics, construction of the cryptococcal genome sequencing strain, and in cell biology, identification of the first fungal density-dependent quorum-sensing peptide and the heteroresistance to azole drugs. So since I cannot cover all these, I'm going to just talk about this in detail, as I said, because this was really the very basic foundational knowledge that was needed for Cryptococcus. Now, when I joined Dr. Bennett's office laboratory in '74, I was given a chance to study crypto. But I had to have a goal first. So my first goal was uncovering the complete life cycle of Cryptococcus by investigating the possibility of heterothallism. Because until this time, excuse me, Cryptococcus was known as a budding yeast, that's all. And discovered the heterothallic life cycle in neoformans in 1975, and then the other species in 1976. And resolved the taxonomic position of Cryptococcus within Basidiomycetes and established the foundation for the phylogenetic study. Now, this is the way the fungus was known from the time of discovery was 1894 until 1975 when I discovered the sexual state. And now this is just a scanning electron microscopy showing that yeast is covered with a polysaccharide capsule. When I found the life cycle, I was not only happy, but whole things start opening up. First, life cycle here. Now, this is, I call Met A and Met Alpha because there were 2 sexes, Alpha and A. I just named it as Alpha and A. There's no reason why it cannot be plus or minus or something else. And when you mix these yeast cells on the media which is conducive for the sexual reproduction, then this hyphal zone, you can see the hyphal zone here, completely different morphology. And this is what it was. First, mating type A and alpha, just bud and bud. But when they meet together, they conjugate and then produce this mycelial form, which is shown, which is shown here, fluffy white area. And the nuclear fusion occurs at the tip of basidia, and then meiosis and meiotic nuclei remain in the basidium, but just keep on doing mitosis and then bud out the species in the long chain. And now this is a scanning electron microscopy. So completely different morphology of asexual and sexual stage of Cryptococcus. How did it affect taxonomy? Well, we already showed that there must be two species. We knew that Cryptococcus was heterogeneous, but not quite as a two species. So the two species concept was emerged after I showed this perfect state. And before this perfect state, Cryptococcus was always identified as a serotype A or D or B or C. And this is all Dr. Bennett's work. Now, when I did the mating, AD mating type produced this kind of a sexual state, whereas B and C serotype produce completely different morphology of spores. And it's really surprising because of 20 strains that he picked randomly, and there were two mating types. And you will be surprised by looking at why I say this. So if you discover the heterozygosism, how did it impact the field? Epidemiological significance of mating type. We just think that, oh, there got to be med-A and med-alpha about half and half among clinical as well as environmental source. What we were so surprised was med-alpha was 99.8%. Med-A is only 0.2%. If you think about the probability of myself getting 20 strains from Dr. Bennett, in which 2 isolates were med-A. Can you imagine? So I calculated, it looked like probability is 1 in 1,500 chances. 1 in 1,500. And so I was very lucky that he selected 20 isolates, among which there were 2 mating type A strain. That was all he had, 2 mating type A strain out of 1,000. Construction of a congenic set of med-A and med-alpha, because when you see that med-alpha predominates, then you're curious about what is med-alpha different from med-A, and why is it so much more common? And next question, of course, you will ask is, what is the role of mating type in virulence? Med-alpha was found to be significantly more virulent than med-A in the animal model. We don't know about human model because there's just so few med-A out there. And genomic analysis of med loci and evolution of fungal sex chromosome project emerged. Mitochondrial inheritance, uniparental inheritance from med-A parent, just like in mammalian case. The mitochondria comes from mother, but mitochondria from crypto comes from med-A isolate. Now, who benefited from all this discovery of heterothallism and built their career on the mating. And Joe Heitman in Duke, he had all this completing the mating type loci, a genetic map of the mating type loci, and pheromone signaling cascades and cellular circuits that required for mating. And he spent probably no less than 10 years to accomplish all this. And Sharon Ling, now in Georgia, but she used to be in Texas A&M, and she discovered the same-sex mating among Cryptococcus and pheromone-independent unisexual mating. And then Dr. Zhu in Canada, he's the one studied the MET-A-dependent mitochondrial inheritance. So all these people built their career benefiting from heterothallic life cycle of Cryptococcus. When this is 2000, so this is long before cryptococcal genome sequencing was established. And when Marvin Karras from Germany was with us, Yun was teaching how to isolate or map the mating types, and we were so surprised that it is at least 50 kb long. And if you look at Saccharomyces cerevisiae, there are only 2 genes, and they're like less than 8 kb. Compared to that, 50 kb, and all the MET kinase cascade genes were sitting on the MET locus. And as I said, and when genome sequencing was done in 2004, Heitman at Duke completed, and both mating type, the mating type locus was 100 kb or 117 kb. It's almost a sex chromosome. About phylogeny, how did they benefit? How did the area of phylogeny benefited from the heterothallism? Now, if you see this morphology, this is the sexual state. Now we can compare about the remaining fungal species in the kingdom fungi. The nearest one was this structure. It's produced by Tremella. That is wood ear mushroom. You know, wood ear mushroom, the black, yellow, white. Now, they all grow on tree bark. So finally, we noticed that this Cryptococcus should belong to Tremella group. And when DNA sequencing became available, phylogenetic tree based on ITS indeed showed that Cryptococcus group and then Tremella species it's in the same order. So now we could place the Cryptococcus in the Tremellales of Basidiomycota. And because of all this difference, phylogenetic study was performed by many people and eventually found that Cryptococcus gatid has 5 different molecular types, Neoformans has 4 molecular types, and then this person, Dr. Mitchell, is in Duke, and Mariana is in Italy, and Buchhout is in Australia, Holland, and Meyer was in Australia. So it's like whole international teams start studying the phylogeny. It's stuck somewhere. Right. Yes, now. So the first goal was discovering a full life cycle. Now that's done. And the second goal was to find the virulence factor. And how do you find the virulence factor? So what I did was dichotomous diagnostic approach of, that time there were only 27 species in the genus Cryptococcus. So their ability to utilize carbohydrates and nitrogen source and their growth ability at 37 degree, and then the phenotype of colony that was grown on the media that has diphenolic compounds. So I made this diagram, dichotomous diagram, and what stood out now, I have circled here, but it doesn't show. The very at the top, you can left, it says colony is brown, lactose is not utilized, and then grow at 37 degree. And I looked at all 27 species, and that's Cryptococcus neoformans was unique or distinctive, distinguishing character is this. Well, I have a problem. Okay, why brown colony on the diphenolic media? Professor Stibe, who used to be working at Robert Koch's Institute in Berlin, and that time, of course, the ecological niche of Cryptococcus neoformans, the best known was the pigeon dropping. So he thought maybe pigeons eat the bird seed, and the bird seed somehow is selectively letting Cryptococcus grow much better. So he extracted the bird seed, put it into the agar, and then plated the clinical yeast. All the candida species remain white, and neoform Cryptococcus is the only one that produced the brown colony, the melanin. And so, remember, 37-degree growth, melanin formation on media, and negative utilization lactose. So I thought lactose is not very important. So next thing we did was made a mutant. That time, of course, there's no genetic manipulation. You do UV irradiation and get the melanin negative, and then leave the melanin positive parent, cross them, and then isolated all the F1 progeny, and there was no question that's Mendelian inheritance of the melanin formation, exactly out of 25 colonies, 12 to 13. And then 37-degree growth, and then capsule. These were by far the essential virulence factor of Cryptococcus. And two fellows worked on these projects with Dr. Polacek and Judy Rose. Both of them are retired now. But this chart shows the wild type and then revertant of white to melanin colony. Revertant are pathogenic, but melanin negative or capsule negatives are almost avirulent, depending on the strain. And this is a mouse study. So we established that biochemical steps in melanogenesis described, and then involvement of only one enzyme, because it was clear that only one enzyme was involved, and that was lactase. We called phenol oxidase. Deposition of melanin in the cell wall, we noticed. And then Peter Williamson, who came here, he's sitting here, but he came here as a clinical fellow, and the first 20 years of his career was totally focused on how Cryptococcus produce melanin and how it's synthesized, what kind of genes are regulating, how is it being shipped, transported to the cell wall, and eventually secreted. All of these pictures, there are just tons of genetic factors involved to make the melanin formation. So I'm glad Peter started working on melanin as soon as we knew it was a virulence factor. And then Casadevall right now at the Hopkins, but previously he was at New York. He produced melanin ghost made from melanized cryptococcus. And this is anti-melanin antibody stained ghost. And the interesting part right now is anti-cryptococcal melanin antibody made in his laboratory is currently in evaluation for the treatment of human melanoma, because human melanin, cryptococcal melanin, are both eumelanins. Okay, discovery of this CGB diagnostic media and the impact on the progress of cryptococcal ecology and epidemiology. Before my media was made, what we knew was just about the natural source of cryptococcus was pigeon droppings, and then the soil contaminated by avian guanos. And second species, Cryptococcus gadi, was never isolated from environment until 1990. So because gadi and Neoformans was discovered just 1 year apart in 1894, one and 1895. And so it's almost like 100 years that we didn't know where gadi was coming from. Our diagnostic media became instrumental for the discovery of C. gadi from eucalyptus tree in 1990, which led to research for arboreal source of cryptococci, because it's natural. Their tremella and cryptococcus are very closely related. The tremella grows on tree bark, and why not cryptococcus? But everyone was focusing on the pigeon droppings. That's why we never found until 1990. So this is media. Neoformans does not utilize, cannot grow on CGB agar, whereas gadi can grow and change the pH of the media. It becomes blue within 24 hours. Using this media, fellow in Australia, Dr. David Ellis, he sampled eucalyptus tree the leaves and florets and tree barks swabbed, and tons of yeast were isolated. But he had to look for gadi. He used my media, and he isolated the first gadi from eucalyptus tree. And then there are hundreds of other people start looking for other trees, of course. So this is ecological impact. But what about epidemiology? Cryptococcus neoformans, at least 70% of them are from HIV-positive patients, clinical source. And then only about 25% comes from others, as I said, what the risk factor in the beginning. But interestingly, if you look at the gadi, very few comes from AIDS patients, and the remainders often say previously healthy patients. So they thought it was immunocompetent patients until we found that GM-CSF autoantibody was a risk factor, which has never been tested in the laboratory when they were looking for patients' immunological panel. And so gadi was found to infect previously healthy patients. So epidemiology and ecology both developed very fast after this media has been formulated. Now, I looked at all the trees from which the Cryptococcus comes from, but I just picked the 12 here because these names like acacia, pine, and oak tree, and platanus, and these are very, almost everyone knows what they are. But there are about more than 100 trees that gadi and neoformans are habiting. And by 1985, that PCR became available. And all of my work so far has been classical, And then the handwriting on the wall of my lab keep on showing that you need to change the research direction, reorient your direction to molecular genetics. It was very inconvenient, almost scary to step out of my comfort zone, but I decided to change it. That was 1985. That time I was already early 50s, and it was really a hard decision that I had to make. And so what did I do after changing the direction? First thing I did was developing the molecular technique of Cryptococcus and come up with the transformation method. And then Robert Koch's, the virulence factor, molecular cause postulate was fulfilled. So I'm going to talk about these two because this too comes after my reorientation. And I have no time to go through here. So shifting research field, what did I do? I enrolled for each molecular biology course offered by Cold Spring Harbor, and I left home and stayed 3 weeks in the lab, and I found I was the only one who are in 50s, and the rest of them were in their 20s and 30s at the most. And so developed the molecular tools upon arrival, come back to lab, and then developed with the collaboration of Jeff Edman in UC San Francisco. And so now we had developed transformation technique developed. And that made us to do molecular cause virulence factor, molecular cause postulate for fungal virulence factor. And by doing cloning capsule genes and transforming cap minus mutant, cap positive disruption, and all of these made when Yun Chang, who is a staff scientist in our lab, took this project and used the cap minus mutant. It looked like you don't see any caps. Colony is very dry, so when he complemented with the genomic DNA, it became not only capsule positive, of course, mucoid colony, but the most important part is all this virulence came back. The greens are the cap minus strains of at least 4 different strains, 4 different mutants. And then the red is the wild type, and blue is the one which is complemented this mutant, and they all became virulent. So after this huge change occurred, Cryptococcus research field experienced a sudden turnabout from mid-1990s. Why? Because AIDS came on, and then low level of lab hazard, so they shouldn't be afraid to move in from other field, and genetic amenability due to the crossing and transformation and all of these, availability of molecular tools, and conformed virulence factors so they can go after virulence, and genetic diversity, and excellent animal models. Because of all these, people start moving in, influx from all these areas with a strong background in molecular genetics of Saccharomyces or Eustachilago or other microorganisms. And glycobiology, because the capsule being the major virulence factor and knew very little about capsule synthesis, so glycobiologists came in, and evolutionary biologists because of the diversity of cryptococcal population, and genetics, bioinformatics, and cellular and molecular immunology, especially because excellent animal models. Eventually, by year 2011, Cryptococcus became a model yeast for the study of fungal pathogenicity, and published a book by ASM. I tried to have as many people's picture, but this is all I could get. And by year 2017, all of these people, I call it village. Plus, there were about 400 authors, co-authors, collaborators, and worked so that I could accomplish to this date. Conclusion. NIID played a key role in laying the foundation for progress in the cryptococcal research starting in 1975, and reorienting the research direction to stay aligned with scientific advancement was inconvenient and challenging, but it was crucial for sustaining a long and productive career. Our contribution transformed what was once considered an obscure pathogen, because there was just 300 cases a year, new cases, until AIDS came in, into a model organism in biomedical research as the HIV epidemic heightened the importance of cryptococcosis. I am deeply indebted to my mentors and dedicated co-workers and supportive colleagues who number more than 400 to date. Thank you.
Looks like we might have a question over here.
Oh, I have a naive question.
Please.
How does the melanin contribute to virulence? Is that known?
Excuse me?
How does the melanin contribute to the virulence?
Many, many different ways. I think that the best way will be Peter Williamson will answer, but it protects the cryptococcus because it's antioxidant, and then, of course, it's accumulated in the cell wall. So once it's phagocytosed, the melanized cells are much more difficult to be killed.
Thank you.
Actually, I have a question for you. You know, I'm thinking back on when I was doing clinical work, and most of the people I saw with cryptococcal meningitis as a neurologist were people with diabetes that was out of control.
Right.
And yet the cryptococcus apparently can live in low glucose environments. Is it the immune system of the uncontrolled diabetic that
Yes.
is this something unrelated to the glucose content of
Yes.
their blood?
Maybe I didn't mention it, but this cryptococcosis is primarily for immunodeficient patients, number one. So I'm sure diabetes patients must belong there too. But the reason why it can thrive in the brain is because this organism can thrive on low glucose media. Not that crypto dislike the high glucose, but they can survive very well in low glucose.
Thank you. Thank you very, very much. Our next speaker is Dr. Thomas Kunkel.
It's a great pleasure to be here. I've never had to give a talk that was described as it was to me to try to cover 42 years of research in 30 minutes. So I'm going to try to do that with minimal new data, but to describe some of the things that have really struck me in my time as a scientist. So I was, as a child, a member of a large family, 11 of us, 6 brothers and 2 sisters and my parents, and we grew up in a tiny little tobacco farm in the hills of Kentucky. And the furthest thing from my mind as a child was to become a scientist. And what led to that was the fact that my father had a heart condition and passed away at the age of 60 when I was very young, and we had to move into the city, and that actually broke my heart, because until I was about 25 years old, I thought I was going to be a farmer, and that was what I dearly loved to do. But at the same time, I enjoyed other things, like very much enjoyed science fiction, and I would read books about science fiction from the time I was about 7 years old until today. And I still enjoy that, and that's illustrated by this to your left-hand side, the Milky Way as it might appear from 10 billion light years away. And that's 10 to the 23rd meters, and we're all scientists and know that that is a huge distance. And on the right is what I do, which is to study the fidelity of DNA replication, and it's illustrated there by the X-ray crystal structure of a DNA polymerase, correctly pairing an A incoming opposite a T. And the distance there is absolutely huge between the left and the right. This is the evolution of life on Earth as presented to me by my excellent colleague, Roel Schopper. The formation of the moon is on the left, and that was 4.5 billion years ago. And that's almost unimaginable for us to think about what a billion years is. And on the right is what the NIH does, which is to understand the impact of the environment on human health. And you can see over there, Homo sapiens has only been around for a very, very, very tiny slice of time. So one of the things that has always intrigued me is, how did life evolve on Earth over 4.5 billion years? So time is a very critical determinant of the answer to that question. This is another image provided to me by another good colleague of mine at the NIEHS, Environmental Health Sciences Institute. We live in North Carolina, so I don't get to walk down your beautiful hallways here. I was very impressed with all of the stuff that you guys probably see every day and maybe don't even pay attention to anymore, which is the list of previous colleagues of yours who won prizes and discovered biology as we know it today. And I took quite a while coming down the hallway here to Lipset to read some of that stuff. Like right outside is Marshall Nuremberg, and Marshall Nuremberg was the mentor for one of my best friends in the world who recently passed away. So I'm not that far removed biologically from the time frame that made NIH great and the basic research that determines life on Earth. Jeff Mueller provided me this. He's an NMR spectroscopist, and this is the tools used on the bottom to discover protein dynamics and macromolecular motions using nuclear magnetic resonance. And the time frame here is very, very different than the 4 billion years of evolution that I described. In fact, if you want to understand replication fidelity, then you need to understand what goes on in the X-ray crystal structure of a DNA polymerase, where the length of a hydrogen bond between an A and a T base pair is 2 angstroms, to roughly the size of a human being, which we could describe as being 2 meters tall. And it covers a time frame of 10 to the minus 14th seconds to greater than 4 billion years. And in order to cover that immense amount of territory to understand DNA replication fidelity, we need 4 basic technologies, which I describe as structural biology, biochemistry, genetics, and genomics. If you want to type the human genome, it's 6 billion nucleotides. And if you were typing on a word processor, you would use selecting the correct letter of the alphabet. You would use backspace, and you would use spell check to ultimately get an error-free document. Now, I had a slide that I took out that was 500 letters, and I had students come into the lab over the years who used 1, 2, or 3 of those methods to type that 500 letters, and there's their error rates. About 20 mistakes for every 500 nucleotides if all you use is the first step. If you add the second step, you improve fidelity. And if you add spell check, you get an unblemished document. Same holds for replication fidelity, where the DNA polymerase that selects the correct nucleotide has a certain selectivity for a correct over an incorrect nucleotide. Proofreading, that is the exonucleolytic removal of a mismatch, is roughly equivalent to backspace, and spell check could be roughly equivalent to 2 major repair processes that I would like to briefly describe in the next few minutes. One is DNA mismatch repair, and the other is ribonucleotide excision repair. The world's expert on that subject is somewhere in this audience listening to me. That's Bob Crouch. And what we have been investigating for the 42 years that I've been at the NIEHS was to try to understand how replication fidelity can be what I consider one of the most amazingly accurate biological processes that occur. And I'm going to give you a little bit about that as a person who was originally trained as a biochemist and picked up structural biology, genetics, and genomics as time went by. So this is what we believe summarizes what it takes to replicate the 6 billion nucleotides of DNA in the nuclear genome of a human being. 1, 2, 3, and 4 are the processes that we have been vigorously investigating for the 42 years that I've been at the NIEHS. And most of that was on the selectivity of the DNA polymerases themselves, number 2, in which you have a correct, properly aligned DNTP incoming opposite a correct template base, and you also have the correct sugar moiety, deoxyribonucleotides instead of ribonucleotides. But over time, we have investigated all of those processes. And I don't have time to describe to you everything that we've learned about those, so I picked a couple of selected subjects that have had an impact on how we think about replication fidelity. And by that I mean how the world thinks about it based on what is now in molecular biology textbooks, partly based on what my lab has contributed to this area of investigation. And the reason we're interested in is the responsible mechanisms that determine replication fidelity. There are connections to human health, which is seminal to what the NIH does as an institution or a set of institutions, and the evolutionary conservation of those processes, which is more of a personal interest on my part, but nonetheless important to human health. And the processes that we think about are the elongation of DNA on an open template without a downstream double-stranded nature. So it's adding nucleotides to a template primer for 6 billion nucleotides and doing it without making a mistake. And then there's Okazaki fragment maturation, which is the process by which on the lagging strand of replication, when short Okazaki fragments are made, you have to get rid of the RNA primer that was used to initiate those Okazaki fragments, and you do that by a strand displacement reaction. And that occurs about 25 million times per replication of the human genome. And then the post-replication repair processes. There are tons of replication or repair processes that occur in cells. They're done by the 17 normal human DNA polymerases, which my lab investigated for more than 25 years. And they occur by mismatch of those many DNA repair processes. 2 of them are particularly important to the fidelity of replication of the human nuclear genome. So these are the 17 DNA polymerases that we know about to date. In my lab, over time, we've investigated the fidelity with which they can replicate DNA without making a mistake. And I'm going to leave most of, or about half of that work behind and focus on one aspect, the fidelity of nuclear DNA replication. And our current work is primarily done in budding yeast, Saccharomyces cerevisiae, as a legitimate molecular model for what goes on in humans. So these are the 3 normal replicases that replicate the 6 billion nucleotides. They're polymerases alpha, delta, and epsilon, or poles 1, 2, and 3. And this slide is just to show you that they're heavily conserved in mammals and in other eukaryotes, including the 2 yeast organisms that we have some information on, Saccharomyces cerevisiae and Saccharomyces pombe. And at the very bottom, you'll see that they have a fidelity of DNA replication done by our biochemical measurements over the years that are different. Pol alpha lacks a proofreading exonuclease, and its accuracy is about 1 mistake for every 10,000 nucleotides polymerized. And delta and epsilon are much more accurate, and they do much more of the replication because the current work suggests that alpha only initiates short chains by laying down an RNA primer and then extending it by a few more nucleotides with DNA, whereas delta and epsilon do the majority of leading and lagging strand replication. So how do we know about the fidelity of leading and lagging strand replication? Well, here's one way to address that question. We can use mutator alleles of those 3 replicases, which have an active site point mutation in what's called leucine 612 in the X-ray crystal structure of yeast pol delta. And that we can make mutant in which we change that leucine to methionine, or its homologue in the B family replicase family in pol alpha is an equivalent leucine that we change to a methionine, or in epsilon, a methionine that we change to glycine. And what we know from years of work on those proteins is that they have preferences. You can make 12 possible mismatches in order to get base substitution errors. And we know that they are selective in making one particular mismatch over another in order to explain a base substitution mutation that occurs in a cell. So, for example, pol delta, L to M, has a 28-fold higher probability of inserting G opposite T than it has of inserting C opposite A when the complementary strand is being replicated. So we can use that kind of replication fidelity asymmetry for each of these mutator polymerases and ask what happens when you sequence a human genome or a yeast genome that's been through 30 passages on a petri dish, which is equivalent to about 900 cellular generations or 900 replication cycles. And what you realize when you sequence the whole genome of a pol delta L to M mutator allele is that the kinds of mutations that appear in the cell depend on where the mutation is and which of the replicases is doing the replication. So in this example, pol delta makes errors on the left-hand side that are consistent with G opposite T, and on the right-hand side with errors that are consistent with G opposite T on the opposite strand. You can use this kind of logic for all 3 replicases and all 12 base-base mismatches and arrive at a model which suggests that pol alpha is initiating Okazaki fragments on the lagging strand. Pol delta then continues to do the bulk of chain elongation of Okazaki fragments, and pol epsilon is the major leading strand replicase. That's a question that has been posed for years, ever since Watson and Crick described the asymmetric configuration of DNA. And the data that we generated on these mutator alleles was consistent with that model on the lower right-hand side. And that's now, I think, you never prove anything in science. You only test hypotheses and try to disprove them. But if you can't disprove them with any observations you make, then you put forward the hypothesis that you believe is true. And I believe that that model defines the roles of the 3 major replicases in replicating the leading and the lagging strand. And that's one of the observations that led to my election to the National Academy of Sciences in May. That's not nearly enough. So we also, in 2010, and this is a statement addressed to any of the students in the audience or the younger students, I'm still a student, but people trying to figure out what they're going to be when they grow up, they might want to think about this. Ribonucleotides are not normally considered part of DNA, but I got a student in the lab in 2009 who worked with my postdoc and found that ribonucleotides, the wrong sugar, not the wrong base up there in green, are frequently incorporated into DNA. In fact, for years, people had thought that DNA polymerases incorporate deoxyribonucleotide triphosphates, but they're very, very poor at incorporating ribonucleotides. Based on X-ray crystal structure, you could imagine why that's so. Based on calculating the DNTP and the RNTP concentrations in yeast cells by our colleague Andre Chavez at Umia University, we discovered that DNTP concentrations are much, much lower than our ribonucleotide incorporations, consistent with much more frequent or much more abundant ribonucleotide synthesis to make the rest of the genetic code work than our deoxys. But polymerases, DNA polymerases, use much lower DNTP concentrations. This can be put into the field of, coupled to the field of DNA repair by the following. This is a slide taken from, given to me by Thomas Lindahl about 20 years ago, describing, Thomas Lindahl won the Nobel Prize for his work on base excision repair, but he was also a very strong proponent of DNA damage and how it affects repair processes, replication fidelity. And this is a list of the lesions in DNA that had been studied for about 50 years at that point, and how frequently they occur in a human cell in the third, or in the second from the right column. So that's the number of abasic sites, which is the most abundant lesion produced in a human cell, at least as far as we knew 20 years ago. And the others are less frequent than that, but they're all highly mutagenic and very, very interesting to people interested in mutagenesis like I am. And so we gave all those numbers over there, little dots, describing the abundance of that kind of damage in a cell. And when my postdoc and her student worked on how frequently the replicases incorporated ribonucleotide triphosphates into the nuclear genome during normal replication in yeast, no damage, that's what we saw. That of all the kinds of non-canonical nucleotides put into the genome in eukaryotes, the most abundant are ribonucleotide triphosphates. So that's led to a lot of work that we and others have done. Now the number of people working on ribonucleotide incorporation during replication is much, much larger than it was in 2010 when we published this result in the PNAS. And we've done two things with it. We've used ribonucleotide incorporation in a yeast cell that could not repair those ribonucleotides. And we found that using this new procedure that we developed a long time ago, hide and seek, when we map where ribonucleotides are put in, they are entirely consistent with our model from the mutagenesis studies, which show that ribonucleotides are frequently put in by pull alpha, less frequently by pull delta, and less frequently still by pull epsilon, but they're still abundantly misincorporated into the nuclear genome. And where they are put in is entirely consistent with our model that's now in textbooks of which roles those three replicases perform during DNA replication. We've also asked what are the consequences of ribonucleotides in DNA. You can go to that annual reviews biochemistry review that we published a couple of years ago, and it describes a lot of biology, genetics, and now genomics that describes how many ribos are put in during replication and how they are repaired, and they are repaired by a process which we now call ribonucleotide excision repair, which is, in addition to mismatch repair, a strong protector of putting the wrong thing into the genome. And when we do that, and I forgot what I did here, if you knock out RNase H2 in humans or yeast, you have lots of ribonucleotides that were inadvertently incorporated during replication, and when they're not removed because of the RNase H2 defect, they cause mutagenesis, which is one of the main things that my lab is interested in. So if you're interested, you can read that article. We're very proud of this work because for 50 years there was biochemists telling us ribonucleotides were incorporated during replication, but only rarely, to in 2010 discovering that they're not that rare. And that implies that there may be some positive selective advantages to incorporating ribonucleotides into DNA, one of which we propose is as a strand discrimination signal for DNA mismatch repair. So this is eukaryotic mismatch repair, and you can see going from the top where a mismatch is in that little hexagonal box, that's left behind by the replication fork, and it needs to be repaired or you get mutations that, among other things, cause cancer. And the steps that involved are a number of proteins ultimately leading to a correct molecule at the bottom in which that mismatch repair has been removed. And so we've been studying that as well for quite some time, since and even before Paul Modrich won the Nobel Prize for studying DNA mismatch repair back in 2015. And we've been studying it based on an article published in the PNAS by Matt Messelson at MIT, who wrote in 1976 that if you want to understand the relationship between replication and DNA mismatch repair in vivo, it may be possible that mismatch repair acts in a directed manner, possibly because of a special relationship to the replication complex. My lab, which is very interested in mechanisms that determine replication fidelity, has been investigating those processes. Does mismatch repair efficiency depend on anything described in black below that? And we've done quite a bit of work on that over the years. And I just want to end by showing you just a couple of things. This is a summary slide of what I've said, where we believe the replication fidelity of the nuclear replicases are those numbers colored in colors up there for base substitutions. We also have done a lot of work on insertion deletion mutations. We also believe that ribonucleotides are abundantly incorporated during replication, that both of those kinds of lesions can be repaired by either mismatch repair or ribonucleotide excision repair, and we published a paper in Molecular Cell a while ago showing that nucleotide excision repair occurs by cleavage by RNase H2 to create a nick in DNA, and that nick can possibly be used to repair replication errors by mismatch repair. That's a summary of what we've done. Now I want to show you just a little bit more. In the course of this work, back in 1985, when I had been running my own show, which was me and one technician to begin with, at the NIEHS, I discovered a method that could be used to engineer any DNA sequence to change its nucleotide composition to make mutations in genes. And I published that in the PNAS in 1985, right after my lab got started. And I put that up there not to brag, although that's one of the reasons that I think I got elected to the National Academy, but it's because within about 5 years I did a literature search, and it had already received more than 10,000 citations. So it's one of the top few publications the NIH has ever published in terms of the number of citations. And that was because that procedure was so applicable to many, many questions in biology where you want to make a mutation in the gene. But it was done in 1985 when it was very difficult to do unless you had some phenotypic selection. And this allowed me to do that as one event out of every 2 genomes that I sequenced without any phenotypic selection. You can read that paper if you're interested. But it made me think very strongly last week when a colleague of mine sent me a publication and said, "You've got to read this." And it was "Applied Research Won't Flourish Without Basic Science." And this was by the 3 authors listed there who described the portfolio of research that NIH has funded since its existence, and decided that about 51% of that research is currently basic research, which is what my lab studies, very basic questions, and about 49% is currently in what I call artificially translational research, as beautifully illustrated by the first talk we heard here. So with that thought in mind, I wanted to end this by describing you one thing that we're really excited about now in my lab. I'm interested in the question of, does simple chain elongation copy DNA with very, very high fidelity, whereas strand displacement, which occurs during Okazaki fragment maturation, requires that an RNA primer be removed from the DNA, and that's done. Well, I'll show you how that's done in the next slide, but going back to one of those slides I showed in the middle, elongation on an open template has to occur for every nucleotide that's incorporated, or most every nucleotide. But Okazaki fragment maturation uses the lower thing, strand displacement. So we're very interested in that question energetically, biochemically, structurally, but we're also interested in that from the standpoint of applying whole genome sequencing, a genomics approach to the question. So we did that in a yeast strain that lacks Okazaki fragment maturation processing because it lacks flap endonuclease 1, which disrupts that flap so that it can be excised by FEN1. And when we did that a few months ago, we found something that totally confuses me, but in a good way. It's always great to be confused because eventually, if you work hard enough on it, you can find out something interesting. So, I'm going the wrong way. I apologize for that. I just want to show you two slides. When we investigated mismatch repair, if I can ever get back to that, one of the nucleases that's thought to be involved in removing a mismatch once the newly synthesized strand has been nicked to include an entry point for a nuclease, one of those nucleases is FEN1, which works during Okazaki fragment maturation. And that's what I was showing here. This, however, is a very complicated slide, so I'm not going to try to explain it too much, but to say that this is the change in length in base pairs, either insertions on the right or deletions on the left, which occurs in a strain that's lacking mismatch repair completely because you've disrupted the first seminal protein in the pathway, the mutase alpha heterodimer. And in green, you see all of the single base mutations created in an MSH2 strain, one of the reasons it's suspected of being heavily involved in tumor formation. And as you go up and out, you see there's much less lower rate of replication errors in a mismatch repair defective yeast cell, as illustrated by the red, which have, if you could read it, and you can't, lower mutation rates. So the predominant consequence of knocking out all mismatch repair is point mutations. And we published this quite a while ago, but I put it like this because this is what happened when we knocked out RAD27. So there are so many things different about this slide than knocking out mismatch repair. It really confuses me, but you still see green, a lot of frame shift mutations resulting from loss of DNA mismatch repair, so RAD27 is involved in that process, but you also see a lot of other errors in various errors around this curve. They include both deletions and insertions. And so we believe that there's at least one other mechanism that is critical that RAD27 flap endonuclease be there for because of the high rate of mutations in other regions of that plot. We don't understand that. I think, as does my colleague who is responsible for this work, that there may be a total of 30 different mechanisms responsible for this, of which all of them are affected by loss of RAD27, which, another way to say that, is it's doing much more than creating point mutations, which is the major effect of loss of mismatch repair. I don't understand that, except, I could go on for half an hour about this, but I'm not going to. I am going to point out that in the last few years, many articles have described mutational specificity in tumors isolated from a wide variety of cancers. Every time I open up Cell or Nature or Science, I read another very exciting article on that subject. These studies focus on the identity of point mutations that drive tumor formation towards defining targets for clinical intervention, the translational side of what NIH does. The results I just showed you on that last slide in the yeast RAD27 mutational profile suggest that such drivers may include genes, mutations in genes that drive larger mutational events. And that subject has not yet been investigated by those multiple exciting translational research studies. So there is a wide opportunity for any of you students in the audience to become involved in this, which is still a growth area. And last, I want to sincerely thank the many outstanding colleagues that I've had over the years for contributing to this research for now 42 years. Thank you.
Again, questions? I'm going to check online. I know there's a lot of people watching, and perhaps from North Carolina.
Hello. Thank you for the interesting talk. I'm just wondering, any perspective regarding to aging and fertility?
Yeah, there are lots of studies in the literature on the role of mutations or failure to correct mutations in the aging process. Very robust set of literature. In fact, NIH has an aging institute, and several of my colleagues in that institute could tell you plenty more about that subject than that brief answer that I just gave.
Is there an analogous process for when a mistake is made in RNA elongation, like in the formation of mRNA?
Yes. That's not a literature that I spend a lot of time studying, but there are a lot of analogies to error correction during RNA synthesis and error correction during DNA synthesis. There's a world of research done on that subject. Of course, one error in RNA synthesis is not as severe as one error in DNA synthesis because you make a lot more RNA than you make DNA, but nonetheless, there are analogies that have been drawn between the two processes.
Great, thank you. All right, thank you very, very much. I've been asked before our final talk just to let you know that there will be a reception after this session right in the lobby directly outside. And our final speaker is Dr. Giorgio Trinchieri.
Thank you very much, and I'm very happy to be here. I'm really excited to hear the previous two talk. I think my talk will have something a little bit different, that my career has been not very stable. I've been moving around, I think, a lot more than the two previous speakers. And I really started in Italy, studying medical genetics with Professor Ruggero Cepellini. Then I moved to the Bachelor in Immunology. I came to the States at the Wistar Institute in Philadelphia. I went back to Switzerland in Lausanne for a couple of years. And then I moved to back to Wistar for 20 years. And from Wistar, I went to a company, Schering-Plough, in the directed laboratory in France. And in 2004, coming back from the industry, I was very happy to come to NIH. First, I did a sabbatical. I would mention a little bit of that, NID, and then for almost the last 20 years in the National Cancer Institute. And so I'm going to try to put together my movie and history with some of the science was going in the same time. And one of my first projects in Torino, but also at the Bachelor in Immunology, was to identify what the cytotoxic T lymphocyte would recognize within the major histocompatibility complex in HLA in human. So obviously, we know a lot now about HLA, class 1, class 2, many different genes, class 3, and we know the involvement in disease and all of that. But that was very different in those years, talk about 68, 72. We knew that there were antigen that were recognized by sera from multiple women, and they were called serological defined. And just at that time, there was discovered that there were actually two different loci, A and B. These are the antigens that were described originally by some of the pioneers in the field, Jean Doucet, Jan van Rode. But also Fritz Bach at that time found that when you mix lymphocytes together, they activate allogenic lymphocyte, they activate the mixed lymphocyte culture, and define a separate locus in the HLA chromosome that he defined as lymphocyte defined, that now we know is a class 2 MHC. So what we did, we set up a mixed lymphocyte culture, which stimulate in unidirectional donor A lymphocytes, stimulated by lymphocyte from a donor B. We knew that was regulated by this lymphocyte-defined locus. But when we looked at cytotoxic T cells that were generated in this culture, they were killing not from the locus in which they are activated, but they were recognized, the serology, the same antigen they were recognized by the antibody, then the class 1 HLA were recognized by the cytotoxic T lymphocyte. So we identified that the T lymphocyte recognized class 1 antigen originating cell. That obviously was important, for example, in transplant, in allogeneic graft rejection. But a couple of years later, Rolf Wingernagel and Peter Doherty have their major finding from which they got a Nobel Prize, that the recognition of viral antigen is actually restricted by self-class 1 MHC. So the cytotoxic T lymphocyte that will recognize virus would also recognize class 1 MHC. But what was not known at the time, there was a lot of discussion, is whether a tumor antigen also needed class 1 for recognition. So when I moved to Worcester, I worked with Barbara North. We made a lot of different mouse human cell line and hybrid that were transformed with SV40, expressed SV40 T antigen. And we find out that if you immunize mice with SV40, syngeneic SV40 transformed cell, they will kill SV40 cell when transferred in vitro, cytotoxic T cell, but only if they were from the same strain we immunize. So B6 mice only kill B6 SV40 transformed cell, BARB-C mice only kill SV40 T cell transformed cells. So we found out that the tumor antigen are also recognized by cytotoxic T cell in a MHC-restricted way, the way that Wingernagel and Doherty described. But we had a surprise, because we are also immunized mice, not only with syngeneic SV40 transformed cells, but also with allogeneic or heterologous, for example, human SV40 transformed cell. And regardless of which cell we use to immunize the mice, still only syngeneic SV40 transformed cells were killed and not allogeneic one. So we basically found that the tumor antigen that express on allogeneic heterologous cell are cross-presented in vivo in association with osteosyngeneic cell, what's now known as antigen cross-presentation. So at that time, now we know about the MHC restriction, MHC recognition, we know how the T cell receptor work, and we know all the molecular detail of this type of interaction, signal transduction. But what I was talking about, those studies were in the 70s. The T cell receptor was really described by Ellis Reinhardt and Tak Mak and Mark Davis in the 80s. So at that time, we really didn't know what was going on, and we have a lot of discussion with Peter Doherty when he was at Worcester Institute, and we come out with this conceptual paper and try to figure out what was going on, and we basically, as this drawing from the paper, we said that maybe that MHC changed the virus, so the virus antigen changed MHC, or maybe there are two receptors. Maybe it's a receptor that see what is in between the two. And actually, the first hypothesis that we have, the possibility the H2 MHC was modified by the virus. We know now it's modified by the peptide. Was probably the one close to what eventually was discovered to be the real recognition. Now, what about cross-presentation? At exactly the same time, the same month, in May 1976, Mike Bevan described cross-presentation. In this case, he was working with the minor histocompatibility antigen. And then, obviously, Mike Bevan and many other, Migorena and Melman and so on, really did all the molecular characterization antigen cross-presentation. And what we know now is that the antigen can be cross-presented by specialized cell types, mostly this dendritic cell, actually mostly one subset of the dendritic cell. But again, when we were doing this study, was in 76, the dendritic cell were actually described by Ralph Steinman in 73 as funny-looking cells in the lymph node of mice. It was not only really after 2 or 4 years after the study I've been discussing now, our study, or Mike Bevan's study, that Ralph Steinman and Michel Nussschneider eventually identified the dendritic cell is the antigen-presenting cell in mixed lymphocyte culture and in antigen-presenting in general. And at that time, obviously, we could not attribute neither I nor my group nor Bevan to dendritic cell. And what we were really thinking was what Emilio Nanue was pushing at that time, that the major antigen-presenting cell were macrophages. And we thought that the macrophage would take up the tumor cell in vivo and then present on their H2 to the immune system of the host. And I'd like to mention Emilio Nanue here because Emilio passed away shortly ago, and actually Emilio, together with Marco Corona, was the one that nominated me for the Academy. So the other, when we recognize that the human cytotoxicity cell recognize HLA class 1, we thought that maybe if we use antibody against HLA class 1, remember there were no monoclonal antibody at that time, there was a serum for multiple women, maybe we can block the killing. And we found out if we are using to try to block completely IgG antibody, we did not really block the killing, but we got more killing, and the reason was that were in our culture, natural killer cells, they were able to do antibody-dependent cytotoxicity. And really brought for several years in my group to study many of the biology of this innate lymphocyte of natural killer cell. So with Daniela Santoli and others, we described, for example, the receptor recognizing immunoglobulin on the natural killer cell was the CD16 or Fc receptor 3, and we discovered that it was a major activator for a natural killer cell function. But we also found out that the NK cell that were known to kill a tumor cell or tumor cell line were also very efficient in killing virus-infected cells, but not the same fibroblasts in shown here in this case were not infected by virus. And when we look at what was happening in the killing of virus-infected cell, we found out that production of type 1 interferon was needed to activate NK cell in killing the virus-infected cells. But when we try to identify which cell in the culture was producing interferon, that was not NK cell, that were not a virus-infected cell, but was actually a very rare cell type in human peripheral blood that we define as apologize, it seems to be frozen. Okay, let's go back. Okay, that we recognize that we define a natural type 1 interferon-producing cell, and the cell type now is very well studied. It is called a plasmacytoid dendritic cell. And we found that cells were the most efficient producer of type 1 interferon in peripheral blood. There were some shared characteristic with dendritic cell, but actually quite different. Here show the morphology of human dendritic cell in the blood versus the interferon-producing cells. And we studied that for a few years, but what was surprising that till about 20 years later, immunologists in mouse were claiming that these cells did not exist in the mouse. They were only present in human. So we went to try, when I was in Lausanne, to try to identify the cell, and we found out that actually they are very similar in the mouse as in human. They are mouse type 1 producing cell, and they have this was defined plasmacytoid morphology, and they are now called a plasmacytoid dendritic cell. And what I mentioned that this really brought me to collaborate what was for many, many years was a very close friend of mine, Christine Byron, that we did, that's a really major player in the NK cell field and the virus infection, and we are happy to collaborate in some of this study. Now, the interferon can induce the NK cell to become cytotoxic, but not really induce very proliferation. But we found a few years later that other cytokines like interleukin-2 or stimulation through the Fc receptor will not only induce the NK cell to become more cytotoxic, will also induce them to proliferate, but most important was induce them to produce a cytokine. The major one is interferon gamma, but also other cytokines, they are listed here. Now, this finding that the resting peripheral NK cell, unlike T cell, could rapidly produce high level of interferon gamma and other cytokine without sensitization was really the first identification of NK cell as a first example of what they are known as the innate lymphocyte type 1, similar to NK cell type 2 and type 3. But the other thing that we were very interested at that time was the fact that NK cells were known to be able to regulate hemopoiesis. They could inhibit hemopoiesis, interferon gamma-producing NK cell could activate macrophage, and some of the cytokines could induce differentiation of myeloid cells. So we find out that interferon gamma was playing a major role in this effect of hemopoiesis, but also two other cytokines, the tumor necrosis factor and lymphotoxin alpha, were all working together with interferon gamma in having this effect of myeloid differentiation and inhibition of hematopoiesis. So we started studying all these cytokines. In this year, the cytokines start to be gene clone and produce recombinant cytokine. Interferon gamma and TNF was available. Lymphotoxin really came much later. So we could not get recombinant lymphotoxin, so we decided that we had to purify lymphotoxin. And a good source of lymphotoxin is from EBV transform human B cell line. So we started purifying lymphotoxin from one of these cell lines, RPMI 8866. And while we are doing that, and we tested in fractionation of the supernatant the present where lymphotoxin was, because we knew that the B cell line also activate NK cell can induce interferon production, we decided to see if we see in the supernatant also ability to stimulate interferon gamma or activate NK cell. And this is really the first two columns we did during purification lymphotoxin. The first column was gel filtration, and we found a broad peak that was containing lymphotoxin and also induced interferon gamma production and stimulate NK cell. But then when we purified by ion exchange chromatography, we could completely separate the lymphotoxin action from the ability to stimulate NK cell to produce interferon gamma. So we define this new activity as a natural killer cell stimulatory factor that was ability to stimulate NK cell, induce proliferation NK cell, induce interferon gamma production. And when we purified, especially works in Michiko Kobayashi in my lab at that time, we find out that there is a band around 70,000 in which this activity was contained. But when this band was reduced, it was actually formed by two chains, one of 40,000, P40, one of the 35,000 or P35. So this was actually not a single chain cytokine, was actually a heterodimer in which the activity was found. So that was very surprising because that, all the cytokines at that time were known to be single chain. But then we actually find out maybe we should not be surprised because IL-1, IL-2, IL-8, IL-10, they are not only cytokine, they are all actually Russian airplane, and all these airplanes are single chain. But the first evolution airplane that was dual engine was the IL-12. And IL-12 was really the first discovered heterodimeric cytokine. Alfort then would found that there are structural sequence similarity when the heterodimer is formed by IL-6 and the soluble form of IL-6 receptor. So the IL-12 was studied by us and many others, and was the molecules characterized, the receptor was characterized, signal transduction from JAK kinase, TYK2 and JAK2. I think you're probably tired this day if you listen to TV to hear about SOTYK2, but it's becoming very famous molecules. And then the signaling through signaling STAT4 that would induce interferon gamma and other activate other molecules. So interleukin-12 ended up to be a cytokine that provide a critical link between innate adaptive immunity. It's produced by mostly by myeloid cell dendritic cell in response to many stimuli, including infection, bacterial, and viral, and it's activating on NK cell and T cell. And in 1993, we worked with Sergio Romagnani, who could actually see that IL-12 is the major molecules that is involved in the polarization of the functional subset of T cell, the Th1 T cell, the T helper 1, that are the T cells that produce interferon gamma. And again, like for cross-presentation, exactly in the same month, in April 93, Anogara and Ken Murphy published exactly the same result in the mouse. And at that time, obviously, Anogara was a major competitor of us, but she actually became a very close friend, and we have for many years a wonderful collaboration with Anne. And one I'd like to mention, actually, Anne was elected this year in the Academy of Science as an international academy member. So when we start looking better at the interaction of the different chain with IL-12, P40, and P35, we actually start seeing there were other chains that were interacting with P40, particularly one is called P19, that now we know this form another cytokine that's called IL-23, that was not really the work of my lab. It was mostly the work of what at that time were my colleagues, Sherin Plau, Fernando Bazan, Rob Castellan, and then Ducroix. They really describe many IL-23 and other cytokine of this family, and this really what has become now is a growing family of heterodimeric cytokine. But the important one is really IL-12 that is induced Th1, and IL-23 that at the beginning was some of the activity IL-23 were assigned to IL-12, but were actually due to the ability of IL-23 to induce not Th1, but mostly Th17, and therefore IL-23 is actually a major role in autoimmunity. The work on IL-12 really started what was a very close for almost 10-year collaboration between Philadelphia and Bethesda with the lab of Alan Scher and many others in Bethesda, as well as Alan alumni like Phil Scott, that was already in Philadelphia at that time at the vet school. And we really were moving. The lab, Alan lab would come in Philadelphia. We were coming to Bethesda at lab joint meeting. And we, in all this collaboration, we really work on effect of IL-12-23 in cancer, in infection, and in autoimmunity. It was really a wonderful time of collaborative work on all these different aspects. And I mentioned to you that IL-12 is induced by mostly myeloid cell and dendritic cell stimulation with pathogen or with bacteria or with virus. And that really brought to the thinking back of what is the work of William Coley with the Coley toxin, in which he was able to treat osteosarcoma with a mixture of bacteria, Streptococcus and Bacillus prodigiosus, and induce inflammation, and in a way was induced remission in some patients, and in what was considered to be probably the first, in the late 19th century, the first form of immunotherapy. And as I mentioned, indeed, colytoxin, any bacterial infection, induce IL-12 and then activate TNK cells, toxic T cells that kill the tumor, and our work and many other lab, Tongajesk and others, really show the role IL-12 in tumor killing. And that's really brought Steve Holwyn wrote a book that at that time was quite popular in the immunotherapy field, Commotion in the Blood, in which IL-12 was defined as the next great magic bullet. That time, this interaction with in the tumor immunotherapy really brought me very close to the Cancer Research Institute, in which I'm really still involved after many years. And the Cancer Research Institute, you may know, was founded by Ellen Coley Nuts. That was Dr. Coley's daughter, shown here a few years before she died when we met. And that really brought many years of many discussions, cross-friendship with Lloyd Old, that was running the scientific committee of the Cancer Research Institute. And so that was the next great magic bullet. So did we cure cancer? Well, actually it did not work. It's great in the mouse. It's considered the most potent cytokine-induced anti-tumor activity, but so toxic that was really never successfully used in the clinics, and there were actually major toxic effect during the clinical trial. There is still work. There's still attempted to use in a more localized target way to avoid the toxicity. We did some work with Steve Rosenberg on that, and on T and CAR T cells, and some people are still working on that. And for example, Rosie Kaplan here at NCI is really getting very nice work with transfecting myeloid cells with IL-12 and use that in therapy. Where IL-12 worked was actually inhibition of IL-12 in autoimmunity. So the antibody against the P4010 IL-12 that would block both IL-12 and IL-23 was approved by FDA for psoriasis, arthritis, and Crohn's disease, ulcerative colitis. And the drug made with these antibodies actually selling more than $10 billion per year. And I don't know if I'm proud or ashamed of that, but it's actually one of the 10 drugs the Biden administration target for price reduction. There's probably this drug is not going to be around for long, because now it's been a specific antibody against IL-23, so much more specific in their mechanism, are being approved by FDA, and most likely they will replace the less specific anti-P40. So in 2004, I want to leave my adventure in Big Pharma when I was working for Schering-Plough, and I was happy to be able to do basic science there, but you don't survive long doing basic science in a big pharma company. And I want to come back in academy, and NIH really helped me in that. And it's two persons I really need to thank for that. One is my old friend Alan Scher, that brought me for a 6-month sabbatical, ended up lasting 2 years in his lab. And Kathy Zoon and Tony Fauci really support that, and I really have a wonderful year and a half, 2 years working in an infection parasite lab, and really enjoying many, many friends, old friends and new friends I met there. And then Bob Wiltrott, that I also knew for a long time, enticed me to move to Frederick and then eventually to Bethesda and being NCI, where now I've been for almost 20 years. So what I was describing to you, work between infection disease and cancer, really start, me and some of my collaborators, they really pushed this idea, like Romina Goldschmidt, and really thinking about learning from the two systems, how to learn from infection disease, how to understand cancer immunity, and vice versa. And really the fact that the in infection is the acute infection, it's activated to eliminate the pathogen, but then you should remove the inflammation, resolution of infection, inflammation, immunity, to go back to tissue homeostasis. In tumor, it's actually the opposite. This mechanism of resolution of inflammation are the ones that induce chronic inflammation, induce favor tumor growth, and block immunity against the tumor. And in tumor, we want to do the opposite. We actually want to reactivate the immune system, reactivate inflammation in a way to kill the tumor cells. And in the last 10 or 12 years, what really my lab really started focusing was in a new aspect in microbiology in infection diseases, really was the role of the commensal microorganisms in our body, is the microbiome. And really we'd be interested in studying cancer as a disease of the meta-organisms. So the organisms like us or any other animal, they are formed by the large host, our own cells, together with the commensal microbe. We know that this microbiome is really important for the survival of the meta-organism. Is important in the crosstalk between the host and microbiota, regulate health, regulate many physiological functions. This includes immunity, metabolism, and neurological function. So we really want to study that in cancer. And we work with many of the investigators of NIH. I would say that particularly with Julie Segra and Yasmin Belkaid, in really trying to push the study of microbiome within the NIH intramurals, a new frontier of research. I think this trans-NIH attempt didn't work very well, but obviously the contribution of Julie Segra together with Heidi Kong and Yasmin Belkaid in the field has really been great and fundamental, and Yasmin eventually became this year president of the Institut Pasteur. Our work together with, in collaboration with Yasmin and on our own, received some interest, and I actually was quite excited when National Geographic reported our work. I never thought that National Geographic would become interested in our work. What was not so nice, that the section National Geographic in which our work was described was not exactly rocket science. And then I remember that my son gave me a present a few years before, and I think he was very prophetic. And he gave me this coffee cup, and I still have on my desk. I think it's a good reminder for how we should look at science. Okay, so microbiome is now seen as a major player in cancer, is a player in a possible initiation of cancer genomic instability, definitely in favoring immune evasion, tumor growth, but also as a major role in regulation of cancer therapy. And was really only, I mean, this has been going for quite a long time, it was really only 22 that Doug Hahnemann finally put the microbiome as one of the cancer hallmark. And so what we did was we studied carcinogenesis of microbiome, but also we want to know whether the cancer therapy could be affected by the microbiome. And we established the gut microbiome, determined the efficacy of cancer therapy, mostly by modulating anti-tumor immune response, major effect in innate immunity, but either directly, indirectly, also adaptive immunity. And our paper, in which the major player were Norio Ida, is not showing this picture, it was already back to Japan, but Amian Duchev and his steward Romina Goldschmidt in 2013, was to show that if you want to cure tumor in mice with platinum compound or immunotherapy using CpG oligonucleotide, you need the presence of the gut microbiota. And within gut microbiota, you need certain species that were favor the response. Nicholas T was actually a few years before in a slightly different situation with adoptive T-cell transfer with TIL, showed that LPS and microbiome could have some role. But at the same time, we published in 2013, Laurence Zigovel has been a long-time friend of mine from the time of, she was in Pittsburgh, the time of IL-12, published in Paris the role of the microbiome in other chemotherapy in cyclophosphamide. And the major interest in the community really come when Laurence, as well as Tom Gajewski, show in mice that the microbiota play a role not only in chemotherapy, but also in immunotherapy with immune checkpoint inhibitors, anti-CTLA-4 or anti-PD-1. And our paper and Laurence's paper is actually listed by Nature as one of the milestones in cancer, that 13, I don't know if that's a good or bad luck. And we continue work now for the last 10 years in mice and in a clinical work. And we study, together with many other investigators, some of the mechanisms in which some bacteria, shown here on the left, may favor the response to anti-PD-1. But what we really focus, and I think will play a major role, is our other type of bacteria, mostly gram-negative, seems to have an inhibitory effect on the response to PD-1, and the question is what we want to target for therapy, the positive effect or the eliminating the negative effect. Now, if the microbiome affect the response to cancer therapy, because we can characterize the microbiome in the gut of the patient, the question is, can we predict where the patient will respond depending from which bacteria are in the gut of the patient? And the problem that the microbiome is extremely different from patient to patient. Each individual is about a couple of hundred or more different bacterial species in the gut, and there are several thousand probably distributed in different bacteria, and each person is really different, and each geographic locality is a different microbiome. So when you try to use a machine learning to predict the response, train the machine learning with the data from one cohort, from one geographic localization, and predict the response in another one, it's completely failed. But if you do train your model using all the different cohorts together, leaving out only one cohort, then things start to be better, and we can get a reasonable high accuracy in the prediction of the response. And there are always a lot of, and this work with these 5 different cohorts here of melanoma patients. And there's really a lot of work in our lab and many other labs to try to define microbiome biomarker for prediction of the response. But the other things is, can we use that in therapy? Can we target the microbiome to improve therapy response? And what we did, and all our clinical work, has really been a wonderful many-year collaboration now with showing here Diwakar Davar and Hassan Zarur in Pittsburgh. And what we were able to show that if we give a fecal microbiota transplant from a patient respond to anti-PD-1 to patients that have completely failed to respond to anti-PD-1, we get a reversion, overcome this PD-1 resistance in approximately 40% of the patients. There are now 3 or 4 studies in other group that shown about the same result. That is very promising. But what actually was happening just a few days ago, a study was reported, a preliminary study was reported by a group in Italy that they had the first randomized trial. This is actually a quite significant effect of the fecal microbiome transplant improving in the response in patients that failed therapy before the transplant. And the other thing that we work and we are trying to figure out other way to change the microbiota. And one way that it's possible to do that is with diet. And together with working with Jennifer Vargo in Houston, we'll be able to show that patients with a high-fiber diet are much more responsive to anti-PD-1 therapy, the patient with low-fiber diet, and we completely reproduce those results in mice and show that the fiber, the diet, is affecting PD-1 response by changing the microbiota composition in the, at least in the mouse experiment. So I've been discussing, like my two colleagues before me, quite many years in science. I mean, I can't really list everybody. So I would like to list each person, each contribution. I don't even have a picture of everybody I've been working with, trying to collect here as many as possible. Some of you may know some of them, especially some of the NIH people. But it's been a long journey, and it's been a really rewarding one. And I really should be really thankful to NIH for this wonderful last 20 years and being able to do this type of work. Thank you very much.
I think you know the routine. If you have questions at the mics.
Yeah, it's interesting to me that your journey has taken you from the endogenous immune system and the cells that mediate it, and that I think we all think of as being there to fight this foe, the exogenous microbial universe, all the way almost full circle to the point where you're studying the peaceful coexistence and maybe collaboration with the microbiome. Is there a danger of the endogenous immune system killing off some of these? I mean, are there disorders that result from seeing the microbiome as foe, or is there something that protects these particular organisms from attack by the human immune
Yeah,
system?
I mean, obviously, we evolve as a meta-organism, ecological system, and we are most of the time pretty good. There are innate and immune mechanisms that regulate that. There are, the barrier, the epithelial barrier always play a major physical role in avoiding that. But sometimes things go wrong, and you have two types of pathology. One is what they're called pathobionts. These are commensals that normally are okay, but in certain situations they might become a pathogen. I think Clostridium difficile is a typical one. We all have Clostridium difficile, but if it grows not controlled, it's become a major pathogen. And the other one is the opposite, when our system becoming too activated against the commensal, and then obviously the major disease you can think in this case is inflammatory bowel disease, Crohn's disease, and ulcerative colitis.
Wonderful, wonderful. I think all of the talks that we've heard today really give us a sense that there's always a next question. I mean, I think especially the work we've just heard, there is something about science that keeps one intellectually curious for a lifetime, and that always makes there be a next question. So thank you so much.
Thank you.
Well, before we go out for our reception, I hesitate to say this is a tradition. This is only the second time I've done this, but last year, Chris actually commented that I did a sort of a summative 2 or 3 sentences that tied together 3 talks that seemed at face value to have absolutely nothing to do with one another. This year, what struck me was the sort of career take-home lessons that pervaded really all three of the talks that we've heard. So I've come up with five sort of, I don't know, life and career lessons that I want to share with you, at least one of which may get me in a lot of trouble. The first is one shouldn't be afraid to stick one's nose where some would say it definitely doesn't belong. I think that is some of the excitement of science. The second is that one should hasten to juxtapose fields that no sane person would ever put together. The third is that you should always check your work, and particularly your spelling, before you hand your work in. The fourth, and this may be the problematic one, is that foreign political interference extends to interleukin naming conventions. And the fifth is, and this is the crux of the whole thing, and again pervaded all three of the talks today, you should use your science to form a broad and deep network of friends and a legacy of impact. So please join us for the reception outside, and join me in thanking our speakers once again.